my name is Diana.
My name is Rebecca.
And today we're going to be presenting on the enzyme pepsin. A quick history: it was discovered in 1836 by Theodore Schwann, and he coined the term from the Greek word pepsis, meaning digestion. It was one of the first enzymes ever to be crystallized by John Northrop, and he did this by using dialysis, filtration, and cooling, and to be studied under X-ray diffraction. After the introduction of new separating techniques like gel electrophoresis and chromatography, the amino acid sequence of both pepsin and its precursor pepsinogen were determined.
So pepsin is an aspartic protease. Protease meaning that it hydrolyzes peptide bonds, and aspartic indicating that there is an aspartic acid residue somewhere in the enzyme that is the catalytic site. There are 326 total residues, 43 of them being acidic residues or carboxylate residues, and only 3 of them being basic residues. This lends it to having a very low pI, which in turn makes the optimal pH range very low at around 2. That is why you find pepsin in the stomach. There are 2 domains, as you can see by the green and blue portion of the figure. There are also subdomains within them that are very flexible, which could possibly be to help the enzyme slightly shift to allow substrate and also inhibitors to bind. It's composed mainly of beta sheets, so there are 6 right-handed helices. There are catalytic sites at aspartic acid 32 and aspartic acid 215. The next slide, you can see it a little easier. The 2 are in the middle. There's 1 on each domain. There's actually a beta hairpin loop, which is just a common motif that reverses the direction of the backbone, and it's most likely to protect the aspartic acid residues from any solvent that might damage them. There's 3 disulfide bridges, you can see by the yellow portions there. Those just stabilize the tertiary structure of the enzyme due to the binding and the strength of the bonds. There are also several salt bridges which also help stabilize the folding of the protein due to their strong ionic bonds. So an enzyme that's very similar to pepsin is chymosin, also known as renin. There are 323 residues. It is mostly comprised of beta sheets. There are 2 aspartic residues that act as the catalytic sites at aspartic acid 34 and aspartic acid 216, and tyrosine 77 protrudes, creating hydrophobic interactions. A lot of those effects are very close and similar to that of pepsin. The main difference is where these two enzymes are found. Chymosin is specifically found in a small amount of animals, specifically young animals. An example of that is young calves have chymosin in their stomachs. And the role of chymosin, as you can tell by the aspartic acid residues, is that it is like pepsin an aspartic protease, so it is actually hydrolyzing the milk proteins and allowing the milk to curdle, which in turn lets calves receive as much nutrients as possible from the milk.
Pepsin is a proteolytic enzyme, meaning it's a protein-degrading enzyme, and it functions primarily in the digestive system by degrading its enzymes. It hydrolyzes ingested proteins at peptide bonds on the amino terminal side of aromatic amino acid residues. So this can include phenylalanine, tryptophan, and tyrosine. Now the gastric juice of our stomachs are kept at an acidic pH of 1 to 2.5, and this acidic environment allows the unfolding of large globular proteins to expose their internal chemical structures, which the now activated digestive enzymes like pepsin can use to cleave. Thus, pepsin's optimal pH is 1.5 to 1.6.
Here's the mechanism of pepsin. Since pepsin was one of the first enzymes ever discovered, it's been studied for a very long time. This is the typical aspartic protease mechanism. You can see on either side of the divots there's an aspartic acid residue, and they're just hydrolyzing the peptide bonds, as you can see there. Pepsin particularly has a preference towards hydrophobic and aromatic residues. It hydrolyzes on the N-terminus side of leucine, tryptophan, phenylalanine, and tyrosine in particular.
Now, pepsin's primary role in our bodies involves digestion. So the presence of food will stimulate the gastric mucosa to secrete the hormone gastrin. And the gastric mucosa is a thick mucus membrane layer of the stomach, and it's an alkaline-based layer as well that contains and releases bicarbonate ions in order to neutralize acidic substances that exist in the stomach. So the hormone secretion of gastrin stimulates the secretion of gastric juice, and that includes hydrochloric acid by the parietal cells and pepsinogen by the chief cells. And gastrin also activates digestive enzymes like pepsin. So the acidity of hydrochloric acid will break down the larger proteins, exposing their internal structures to the now activated digestive enzymes. And pepsinogen is converted to active pepsin by autolytic cleavage. So pepsinogen is an inactive precursor, or a zymogen, and is converted to pepsin upon the release of gastric juice. So many proteolytic enzymes of both the stomach and pancreas are also regulated this way.
A little more in depth on pepsinogen being converted to pepsin. As Diana said, it is an inactive precursor that is secreted by the chief cells of the stomach, and that are released due to the hormone gastrin. As you can see on the figure, there's 44 extra amino acids there's an extra 44 amino acid segment that is covering the aspartic acid residue catalytic sites that are buried within pepsin. So pepsinogen can be activated by HCl, hydrochloric acid, which creates an acidic environment and allows pepsinogen to unfold and cleave itself. This is done by the protonation of carboxylate groups. This weakens the N-terminus segment and it leads to pepsinogen cleaving itself. Once the 44 amino acids are cleaved, the active sites are exposed and substrate can bind.
So other uses of pepsin include the commercial pepsin used as a component of rennet, which is primarily used in the curdling of milk. So it acidifies milk, bringing down the pH, and will cause denatured milk proteins to clump together. So primarily, a group of phosphoproteins called casein will clump together. And it can also be used to isolate FAB fragments from antibodies. And FAB is just the antigen binding portion of immunoglobulin. And what it will do is cleave heavy chains near the hinge region. So it's essentially cleaving the disulfide bonds. And this can be used to determine FAB structure complexes more rapidly and also more closely look at the mechanisms of the FAB fragments in the immune system.
One of the journals that we looked at for this project was Local Synthesis of Pepsin in Barrett's Esophagus and the Role of Pepsin in Esophageal Adenocarcinoma, which essentially is answering the question, how much damage does non-acid pepsin cause in the esophagus? Just a little bit of background information. Barrett's esophagus is the damage to the lower esophagus that results from reflux. It leads to replacement of esophageal cells in the bottom of the esophagus into tissue that is similar to the intestinal lining. It's associated with a higher risk of esophageal cancer and requires constant checkups for dysplasia, which are just precancerous cells. Esophageal carcinoma has the fastest rising incidence of any cancer in the US. It has an average 5-year survival rate of only 17%. Endoscopic screenings are done based on the severity of the gastroesophageal reflux disease symptoms, such as heartburn and regurgitation, which occur due to the acidic components of the reflux. So patients often use protein pump inhibitors that relieve the symptoms and not the reflux itself of the gastric content. So non-acidic reflux is still occurring. Past studies have indicated that those taking proton pump inhibitors have a higher incidence of esophageal adenocarcinoma, which suggests that the non-acid components might be more likely to be causing the cancerous cells. Non-acid pepsin has also been shown to promote tumorigenesis, and this study uses all of that information to investigate the presence of esophageal pepsin in patients with reflux attributed to metaplastic conditions from Barrett's esophagus and examine the potential of pepsin to create inflammatory and carcinogenic changes within the esophageal cells.
So 8 biopsies of BE tissue and neighboring normal tissue were collected from the 8 BE patients, and this was done so via elective general surgery procedures like gastric bypass, incisional hernia, or sleeve hysterectomy. So the biopsy specimen underwent Western blot, STS phage, and RT-PCR for pepsin and pepsinogen analysis. So immunohistochemistry was utilized to confirm pepsin protein presence in BE and its absence in neighboring normal tissue. So esophageal biopsies were fixed in formalin, paraffin, embodied glass slides. Human esophageal cells were also collected from healthy patients and treated with pepsin and acid-pepsin, and from that they performed interleukin-1 beta assays in pepsin-treated and control esophageal cells. Now, interleukin-1 beta is a cytokine involved in chronic inflammation and cancer, and this was done in order to determine if the non-acid pepsin exposure could induce the production of interleukin-1 beta cancer-related signaling pathways. For instance, COX-2 reactions are also closely associated with the production of interleukin. And furthermore, increased production of interleukin-1 beta positively regulates the gene expression of PTGS2, which is associated with chronic inflammation and EAC prognosis, so carcinogenesis.
On the left, we see a figure labeled A and B. A is the STS page results, and B is the RT-PCR results. It only shows a shortened figure. They actually studied 8 total cells of each. Here they only show 2 total cells of each. But in the STS page, we see that that pepsin was present in both of the Barrett specimens, and in fact it was present in all 8 of the Barrett specimens, while only 4 of the 8 normal specimens had a pepsin in them. And then we can see from 1B, they were studying pepsinogen A mRNA, which is just the mRNA responsible for pepsinogen, was observed in 2 of the Barrett specimens, but was not observed at all in the normal adjacent samples. And figure 2 was the immunohistochemistry, and then we see here that the Barrett's esophagus had pepsin present and the normal did not.
Now this is an STS page that was ran to see, to look for the presence of pepsinogen in BE patients of the BE tissue and neighboring normal tissue. So as you can see, pepsinogen was present in the Barrett patients, B meaning the BE tissue, and little to none was found in the neighboring normal tissue. And this tells us that pepsin-mediated elevation of cytokine interleukin-1 beta and gene expression of PTGS2 and the COX-2 reactions were observed in the esophageal cells in vitro. So essentially you see a 2-fold increase of the expression of the cytokine interleukin and the expression of the gene PTGS2. And that essentially tells us that with the introduction of pepsin, you're having an increase in these factors that are closely associated with carcinogenesis.
So the results indicate that pepsin was observed in all of the Barrett's esophagus patients. Both the protein and mRNA of pepsinogen was observed in the Barrett's esophagus patients as well, although not all. But this suggests possible local production of pepsinogen. While acidic contents of gastric reflux may promote esophageal adenocarcinoma, non-acidic enzymes like pepsin can also play a large role in the esophageal carcinogenesis, which indicates that we need to study this further and create medication that isn't just treating the symptoms of the acid reflux, but is also creating symptoms of the reflux in general and stop the reflux from happening at all. Thank you.